Welcome back, AP Biology students. Today we're going to be looking at my favorite part of cellular respiration, and that's oxidative phosphorylation. It's a really amazing process. So when we look at our curriculum framework, some things pop out or stand out to us. One is that we're going to be looking at the electron transport chain. You need to remember its role is that it's going to capture that free energy using those coupled reactions we've talked about. And it's going to do all that by establishing an electrochemical gradient. So by the time we finish today, those things should seem fairly clear to you. And I hope this diagram is starting to feel very clear to you as well. So if you haven't already done so, find it, get it out so we can take a few more notes as we look at oxidative phosphorylation today. We have already looked at the process of glycolysis and how the pyruvate from glycolysis has to be oxidized before it can enter into that citric acid cycle, where we end up giving off a whole lot of carbon dioxide as a waste product, making a little bit of ATP, but more importantly, generating those hydrogens that are going to be carried over to oxidative phosphorylation. So today, we'll center our attention on this process of oxidative phosphorylation. We'll look at the role of electron transport in the process of chemiosmosis. So we'll begin by looking at a video summary of this whole process. Once the video is complete, then we'll go back through and look at the individual steps and stages that lead to this massive amount of ATP production through oxidative phosphorylation.
Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Our curriculum framework focuses on your understanding of the role of FADH2 and NADH as those electrons are passed through electron acceptors and eventually ending up in our terminal electron acceptor, oxygen. So as we look at this process, I want you to pay particular attention to the role of oxygen. So the curriculum framework also mentions the proton gradient. This gradient is established in the mitochondrial membrane. However, if you've already studied the process of photosynthesis, many of the things we talk about today may sound very familiar because a very similar process occurs in the chloroplast during photosynthesis. Basically, we're going to see how the high proton concentration is established and how the movement of electrons by passing through the membrane from an area of high concentration to low can allow for the production of ATP within these individual mitochondria. As you saw in the video, the electron transport chain is a set of proteins that are embedded within the inner mitochondrial membrane. These proteins are large globular proteins, and they transfer electrons from one protein to the other. The electrons come from hydrogen, that of course has a proton and an electron. The electron is given to the electron transport chain. The proton is going to be moved to the opposite side of this membrane and help to establish this proton gradient that's so useful. These electrons are going to be passed down the electron transport chain, and ultimately they're going to be captured by oxygen. Oxygen will combine the hydrogen protons with electrons and the oxygen molecules and form water. Once formed, this water could be used for other metabolic pathways within this individual cell, or it might move completely out of the cell and be absorbed by the bloodstream, for example, and then eventually processed through the kidneys and be part of urine formation. Now that the electrons have moved down the electron transport chain and ultimately been captured by oxygen to form water, we still have the proton portion of that hydrogen atom to deal with. So the electron was accepted by oxygen. The proton, though, is going to be pumped to the other side of this membrane, and these hydrogen ions are going to stockpile on the opposite side of this membrane. Notice as this process continues, we're having an increase both in the concentration of the hydrogen protons. We're going to have a charge difference, and we're also going to have a pH difference result because we have these hydrogen ions stockpiling on one side of the membrane. So our pH is going to drop. Our charge is going to increase, and the concentration is going to increase. And ultimately, that threefold gradient is going to produce a proton motive force. Proton motive force is what eventually is going to allow us to produce these high levels of ATP using this unique and unusual enzyme slash transport protein called ATP synthase. These ATP synthase molecules are found embedded inside this inner membrane, the inner mitochondrial membrane, and so there they will establish this gradient from one side to the other of the membrane and create that proton motive force. So we've established a high hydrogen ion concentration on one side of the membrane. When that hydrogen proton moves back through ATP synthase, then that exergonic flow is what drives the phosphorylation of ADP, adding that inorganic phosphate, and that's going to produce or form ATP. And so this is an example of chemiosmosis. A closer look at ATP synthase reveals that this integral protein has two distinct portions. The top part, the rotor, is where the hydrogen ions can come through, and as they do, this exergonic reaction provides the energy needed to phosphorylate ADP. So the ADP molecule gets energy and phosphate, forming ATP in a very efficient method, allowing us to have a much higher rate of production of ATP than if we were trying to do this with just an enzyme alone. And so here's a really good example of how structure matches function in this particular membrane. So we have this unique and very adapted integral protein serving as an ATP generator from the exergonic release of energy fueled by this proton motive force when the hydrogen ions move from one side of the membrane to the other. This picture gives us a really clear summary of some of the terms that we've used during this explanation. The electron transport chain includes all of these embedded integral proteins, and the process of chemiosmosis, where we're using the movement of chemicals through a membrane, occurs at the site where ATP synthase is located. So together, both the electron transport chain and chemiosmosis are together referred to as oxidative phosphorylation. And remember, the oxidative part comes from the fact that oxygen serves as the final electron acceptor at the end of this electron transport chain. So I hope you see how the proton motive force is the direct result of the structures of the membrane. The membrane is built specifically for this job. The mitochondrial inner membrane plays a role in establishing this proton motive force because of its structure. Can you name 3 structural features that make this mitochondrial membrane very effective at its job, what would 3 features be? 1, this phospholipid membrane is impermeable to charged particles. Because it's impermeable, then those hydrogen ions will stockpile on one side of the membrane and they can't just leak back through or diffuse back through. So because it's got this phospholipid bilayer, it is impermeable to charged particles or to charged ions. A second feature was that ATP synthase. ATP synthase is pretty amazing. Remember, it had 2 different portions. It had the transport end and then the phosphorylation end, where hydrogen ions could move through one portion, and then at the bottom portion of it is actually the enzyme-like end of it, where ADP can be joined with a phosphate group and make ATP. A third feature that's likely on your mind is the electron transport chain. The electron transport chain is also a set of proteins that are able to transfer electrons from one another until they're ultimately passed on to oxygen. All right, so now let's consider energy transfer through this whole process of chemiosmosis. Can you arrange these in order? Like, how was the energy transferred? Where does it come from? Where is it going to go? So first, we had the energy in glucose. The energy from glucose was transferred to NADH. We could have also had FADH2 listed here as well. The electron transport chain is where the energy is going to go next. We're going to establish this proton motive force as those hydrogen protons are stockpiled on one side of the membrane. And then ultimately, that energy is going to be transferred to ATP through the process of phosphorylation. Let's look at your summary diagram one more time. I want to make sure that you've put 2 ATPs underneath your glycolysis block and 2 ATPs underneath your citric acid cycle. And then we have an approximation of about 26 to 28 ATPs being formed through that process of oxidative phosphorylation. So you can just see simply by the numbers how much more efficient the process we've talked about today, this oxidative phosphorylation, how much more efficient it is at the process of transferring the energy. Oxidative phosphorylation has a very logical name, in my opinion, because oxidative reminds me of the use of oxygen, and that phosphorylation reminds me that ADP was joined with phosphate to make those ATPs that we are producing at such much more efficient levels, and we used electron transport to do that and the process of chemiosmosis. So you can see how important all the hydrogens are, the hydrogens that were used to stockpile and build up that proton motive force came from all the previous stages of cellular respiration that we've talked about, and played a really important role then in establishing that proton motive force that was used to fuel the generation of this ATP. In this next video clip, you're going to see the process described once again. I would like for you to listen to it. You'll hear the narrator use a lot of specific names for the electron transport proteins. Know that you don't have to know the exact names. You need to be able to describe the process. And so by watching this alternate view, it will help you further develop your understanding of the process of ATP synthesis.
In glycolysis and cellular respiration, a cell breaks down a molecule of glucose and uses its energy to form 32 molecules of ATP, an important cellular energy source. Most of this ATP is generated in mitochondria during the final phase of cellular respiration, during which electron transport and ATP synthesis take place. A mitochondrion has 2 membranes, an inner and an outer membrane, that divide the organelle into separate compartments. The inner membrane contains the components required for electron transport, called the respiratory chain, and ATP synthesis. The intermembrane space comes to be filled with protons pumped from the matrix by the action of the respiratory chain. NADH, along with FADH2, which is not shown, fuels the respiratory chain. NADH molecules carry high-energy electrons that were removed from glucose during glycolysis and the citric acid cycle. The respiratory chain consists of a series of electron carriers, each of which holds donated electrons tighter and with greater affinity than the previous carrier in the chain. When NADH donates electrons to the first carrier, NADH converts back to NAD+. The respiratory chain is important in part because it regenerates NAD+, which is required in glycolysis and the citric acid cycle. The electron transfer to the first carrier, called NADHQ reductase, drives the protein complex to pump protons across the membrane. NADHQ reductase then passes the electrons to a mobile molecule called ubiquinone. In turn, ubiquinone passes the electrons to cytochrome C reductase, a transfer that is coupled to the pumping of protons into the intermembrane space. Cytochrome C reductase derives its name from a redox reaction that it participates in. In redox reactions, molecules that gain electrons are said to be reduced, while those that lose electrons are said to be oxidized. Cytochrome C reductase reduces the next carrier, cytochrome C, by donating electrons to it. The next carrier, called cytochrome C oxidase, oxidizes cytochrome C by taking electrons from it. During the electron transfer, cytochrome C oxidase pumps protons across the membrane. The electrons occupy a relatively low energy state in cytochrome C oxidase, but can occupy an even lower state by joining oxygen and protons to create water. Oxygen is electron greedy and serves as the final resting place for the electrons originally donated by NADH. Without oxygen, the complexes in the electron transport chain would be stuck with their extra electrons, and the chain would be blocked, unable to transport electrons from new NADH molecules. The process of transferring electrons along the chain releases a great deal of energy, which has been used to pump protons into the intermembrane space. This active transport of protons sets up an imbalance of both concentration and charge across the membrane. The imbalance represents potential energy. The protons have a tendency to flow down their concentration and charge gradients. However, they can only flow by passing through a channel in a protein complex called ATP synthase. As protons flow through this channel, they drive the ATP synthase to force together ADP and inorganic phosphate. ADP and inorganic phosphate are both negatively charged, and it takes energy, such as that embodied in the proton flow, to bring them together into a single molecule of ATP.
So as you watch this next video clip, it's going to describe methods that were used to verify that the proton motive force exists. So it's interesting to see how the pH could be manipulated, or the charge could be manipulated to show that this mechanism works with the detail that we've seen.
In 1961, Peter Mitchell proposed a mechanism called the chemiosmotic mechanism to describe how the mitochondrion fuels ATP production. He proposed that a gradient of protons forms across the inner mitochondrial membrane, and that the movement of protons down this gradient into the matrix provides the energy to produce ATP. What experiments supported this hypothesis? In one experiment, the investigators manipulated the pH of the compartments on either side of the inner mitochondrial membrane. In this example, which side of the membrane would represent pH 7 and which would represent pH 8? Because a higher concentration of protons results in a lower pH, the solution above the membrane represents pH 7, and that below the membrane represents pH 8. The investigators began by immersing isolated mitochondria in a medium at pH 8. All compartments eventually equilibrate to pH 8, including the matrix and the intermembrane space. These mitochondria were starved of a food source, so they were unable to create their own proton gradient using the electron transport chain. The mitochondria were then moved to an acidic medium with a much higher concentration of protons. The outer membrane is freely permeable to protons, which allows protons to move quickly into the intermembrane space, lowering the pH of this compartment. These conditions quickly set up a proton gradient across the inner mitochondrial membrane. Protons tend to flow from high concentration to low concentration. However, they only flow through specialized channels in the inner mitochondrial membrane. With the formation of this artificial proton gradient, the mitochondria formed ATP. What is the role of the proton channel, ATP synthase, in the production of ATP? To answer this question, investigators created artificial vesicles to mimic mitochondria. To these vesicles, they added a light-driven proton pump called bacteriorhodopsin from bacteria. When illuminated, the pH of the external solution increased, meaning that protons were pumped into the vesicles. The proton concentration became higher inside the vesicles than outside, creating a proton gradient. If ATP synthase from the mitochondria of cow cells is also incorporated into the vesicles formed ATP.
Do you remember our original questions as we end this unit now? We talked, first of all, I asked, why do we have to eat? I hope now that if you were answering that question, you would be able to talk about energy transfer, that we're eating to provide the energy that's needed so that it can eventually be transferred over to form ATP, which is the cell's energy currency. Also asked you, what does that have to do with entropy? Remember, entropy is that disorder. You are a very orderly system, and so in order to maintain your system and the order of your system, we have to supply a continual source of energy, and that energy has to be in a useful form. And so our cells are getting their useful energy through ATP. And so those two are very closely tied together, that we eat to move the energy into ATP, and the ATP then is used to help defeat entropy. And our third question was, why do we have to breathe? And I hope that you remember very clearly that we breathe to bring in the oxygen, and that oxygen is going to serve as an electron acceptor in the end of that electron transport chain. We have one last topic left in cellular respiration, and that will be to look at the conditions or occasions when oxygen is not available and doesn't serve as the electron acceptor. What pathway can be followed in those cases?